Hello, bachcho. So let's read the question. The question says binding of the specific protein on regulatory DNA sequences can be studied by means of. First option is ultracentrifugation. Second option is electron microscopy. Third option is light microscopy, and fourth option is X-ray crystallography. So the key concept for this question is X-ray crystallography. X-ray crystallography is basically a technique to determine the molecular and atomic structure of crystals. Basically, इसका process ऐसा होता है कि when X-rays are diffracted by the X-rays are X-rays are diffracted by the crystal. जब क्रिस्टल पे X-rays डाले जाते हैं, they are diffracted by the crystal, and इनके जो angles and intensities of the beams ये एनालाइज किया जाता है. फर्दर इसका एक 3D रिप्रेजेंटेशन बनाया जाता है. 3D representation of the electron density within the crystal. And further, इसके जो applications हैं, firstly, it is used to study the structure of DNA. Study the structure of DNA. Second, it basically reveals arrangement of electrons of basically atoms. Electron density के through ये atoms का arrangement बताता है in proteins, DNA, etc. This was basically used by Crick, Wilkins, and Watson to discover the double helical structure of DNA. They discovered the double helical structure of DNA by using X-ray crystallography. So coming back to the question ने पूछा है कि binding of the specific protein on regulatory DNA sequences can be studied. It cannot be studied by ultracentrifugation. Ultracentrifugation basically it separates the particles. It separates the particles on the basis of density and mass of the particle. इलेक्ट्रॉन माइक्रोस्कोपी से भी ये हम नहीं ऑब्जर्व कर सकते बिकॉज इट रेगुलेटरी डीएनए के साथ एक प्रोटीन बाइंडिंग इज नॉट इवन विजिबल इन द इलेक्ट्रॉन माइक्रोस्कोप. लाइट माइक्रोस्कोप तो बिल्कुल ही नहीं. बट एक्सरे क्रिस्टलोग्राफी जब उसको 3D इमेजिंग हम करते हैं, 3D स्ट्रक्चर वो देता है. 3D स्ट्रक्चर्स में से we can determine whether the protein is interacting with the DNA or not. So the final answer for this question is option D. Hope you understood well. Best of luck.